Hello, everyone, and welcome to today's live broadcast, Automated in Vivo Efficacy, Assessment in Preclinical Lung Disease Models, presented by Dr. Laura Schaevitz, Chief Scientific Officer, Vium. I'm Alexis Carls of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Vium. For more information on our sponsor, please visit vium.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Laura Schaevitz. Laura Schaevitz has more than 15 years of experience with in vivo animal studies. As VP of Chief Scientific Officer at Vium, she oversees the development of animal models and draws upon her experiences as a molecular and behavioral neuroscientist to direct the application of Vium's digital metrics to the animal models. In past experiences, Laura utilized a broad spectrum of mouse models, including transgenic and nutrition-induced models of neurodevelopmental disorders such as autism, schizophrenia, Rett syndrome, and blindsight. Laura received her PhD in neuroscience at Stanford University in 2006. Dr. Schaevitz, you may now begin your presentation.
Thank you so much. And thank you for joining me today. I'll be talking about the Vium platform and how it can be used for automated in vivo assessment in preclinical lung disease models. But before we get into the specifics, I want to talk a little bit more about the broader context in which we're looking at this platform. So this is something that many of you may be extremely familiar with, but the cost of drug development is rising exponentially. Here I'm showing you some data from the Tufts Center for the Study of Drug Development, in which you can see that the average preclinical and clinical expenditure of drugs from the 1970s all the way up until more recent 2010 timeframe, where we've gone from somewhere around 300 million for the cost of the development of one drug, up to now it's estimated to be something like $2.5 billion for one drug. And part of that has to do with overall clinical success rates, which are currently down to 11.3% from 16.4% in 2005. And many of those are due to lack of clinical failures due either to lack of drug safety or efficacy, together thought to be associated with maybe about 75% of the drug failures that occur. And so there's a real push for understanding why clinical failures are happening and how we can improve, both from a preclinical as well as from a clinical standpoint, the probability of success. So there's been a lot of work to reduce the number of drug failures, looking at various reasons why drugs fail and potential areas of improvement. Some of those, specifically in the preclinical field, are looking at improved target identification and validation. An area that we're very focused on is improving clinical translatability. Can you create better preclinical models which are more predictive of disease and potentially whether or not a drug would be effective in the clinic? And can you identify biomarkers that may better translate to the clinic? And a biomarker can be anything from molecule to, in this case, we're actually thinking about digital metrics as being potential biomarkers that could translate into the clinic. And another area that people are focused quite a bit on is precision medicine. So can you improve patient selection? And in this case, looking at animal models and understanding potential genes and molecules that would help you better predict whether or not a human in the clinic would respond positively or negatively based on preclinical data. So with that as a backdrop, today's webinar is really around looking at one potential biomarker, in this case, lung function. And can we better build models to predict efficacy associated with drugs and or safety side effects associated with drugs that could translate into the clinic? So with that, I'd like to go to our first poll question. For those of you who are conducting rodent respiratory studies, what are some of the biggest challenges that you've faced? Please check all that apply. These might include their invasive procedures, in which perhaps you can only do a single assessment. Your results may be inaccurate or inconclusive. Perhaps there's a high mortality rate, or maybe you have data gaps or losses, so you only get a little bit of data here or there, not enough to make appropriate assessments, or other. I'll give you just a moment to respond to the survey. Thank you so much for all your great results. So lung function in particular is quite challenging to assess in mice. A lot of studies have been done in the early 2000s, 2010 timeframe to look at various ways that you can assess lung function, basically in vivo in rodents. And here are just a few examples of articles that were published talking about the various challenges and how you might potentially be able to assess lung function or lung mechanics. And there are a couple of methods that researchers can often use, which I'll talk about in the next slide. But really, as we'll see, many of them have trade-offs that need to be made. And this is part of what has been termed by Bates and Irwin as the phenotyping uncertainty principle. So it's the idea that you may be able to get very accurate measurements, but in an animal that is in a non-natural state versus less precise measurements in animals that is in a more of a natural state. So let's look at those. So the trade-off here is between precision and the natural environment. So on the precise side, we have the forced oscillation technique using the flex event. So here it's highly invasive. A mouse or a rat is intubated, and then there's a highly controlled system, which in essence pushes air into the animal, and there are measurements of flow. And this is often a terminal procedure, although there have been improvements in the ability to actually wake up and recover animals from these. But because the animals are anesthetized, they're generally in a very unnatural state. And so you may have abnormalities in what you're measuring just based on the fact that they are anesthetized. But as I said, you get very precise results. And then on the other side, you have data collected in the plethysmograph. So here an animal is placed into a small chamber in which you can measure changes in pressure relative to the outside world. It's non-invasive. You can put the animals in as many or as few times as you'd like to measure them over the course of a study. But the environment is very uncontrolled. And for those of you who have ever worked with this, you know that an animal that is awake and moving around, you'll get results that are quite different from an animal that is asleep and breathing quite naturally. And therefore, the results can often be inconclusive. So before we move to the next slide, I'd quickly like to ask for one more survey question. And this time, asking about the various methods that if you are looking at lung function, you use. So here, the question is, how do you monitor lung function now? Do you use a plethysmograph? Do you use the forced oscillation technique? Perhaps you may use a video-based system, or it's too challenging, and so therefore you don't monitor lung function in your studies right now. I'll give you just a few moments to respond to this question. Thank you so much for those great results. So what I want to introduce to you is this idea that you can actually potentially use an automated home cage monitoring system to get better results or better prediction of lung function in a non-invasive, freely moving animal. So I'll introduce the Vium system in just a second. But as I go through this presentation, some of the questions I'd like you to keep in mind are these results consistent? And most importantly, do they recapitulate something about the underlying biology or pathology? So one thing that we know about using the plethysmograph or using the FlexEvent system is that these often very nicely replicate what you can see with biology or pathology. So changes in the lung function from those measurements will highly correlate to what you might see in changes in the lung. So what is the Vium system? So here's an overview of the platform. The Vium system is a smart housing rack. Oops. So the Vium system is comprised of a rack with individual ventilated cages. Above each of the individual ventilated cages is a white box, which you can see in the center. We call that the slab. And the slab contains video data, HD video cameras, as well as gas pods both in and out so we can monitor temperature and humidity at all times. What I want to focus today on, however, is our use of the HD video camera to actually monitor resting breathing rate and motion in the animals 24/7. So to the very right, you can see a viewpoint from the camera looking down into the cage. You can see an animal off to the very upper left-hand corner there, a black animal sleeping underneath the food hopper. Inside the cage, we also have running wheels seen on the lower left, ladders seen across the upper portion of the screen, as well as water in the lower right-hand corner of the cage. As you can see, the cage is laid out in such a way that there is no place that the animal can get away from the camera. So we've got a good view of the animal 24/7, and we can actually, in what I'll show you in just a second, monitor breathing rates in the animals as long as they're resting. So how do we do this? So here's a slide just giving you a high-level overview of how we create our digital metrics. So in the upper left-hand corner, you can see a video of a mouse, 2 mice in the cage, and there are small green dots overlaid across the video. We actually use complex computer algorithms to compare the location of pixels from one video image to the next. We capture 24 frames per second, and we look at the vectors in terms of the location of that pixel from one to the next to identify vectors of motion, and that's read out on our motion metric. Those same changes, we look for oscillations, and in the spectrum of breathing rate, so between generally 60 and 600 breaths per minute is what we're looking for, and we can read those out as resting breathing rates. going to the next slide here, those resting breathing rates are then read out on the application, which is available to researchers 24/7. And just to prove that the breathing rates that we're getting are very reminiscent of what you would see using more traditional measures, in this case, the plethysmograph, what I've shown on the far right-hand side here is data where we put C57 black 6s as well as C3H HEJs into a cage. So these are multiple readings from multiple animals. We put them in the plethysmograph inside of the cage, and we monitored the breathing rate based on the computer vision versus what we monitored in the plethysmograph. And you can see that there's an extremely high correlation between the breathing rates in both of those instances. So I believe that the R squared here is 0.98. So we're getting a very accurate measurement using this camera-based system to monitor resting breathing rate. So today I want to present 3 use cases. And I'm presenting 3 different use cases with different underlying pathologies to give you a sense for how the resting breathing rate can predict lung function, not just in the instance of one underlying pathology, but across multiple different examples. So here I'll show you data from an idiopathic pulmonary fibrosis model. So we're using Bleomycin-induced. Here we have a very early localized acute inflammation response, and then later on we get fibrosis in the lung. And we can show different results during both that initial acute inflammation phase as well as the later fibrosis. We're also looking at a pneumonia model in which we can monitor this acute systemic inflammatory response. And finally, I'll show you data from a lung metastasis model. We specifically use the colon cancer line CT26 here, and we're showing that as you have increased tumor burden in the lung, it appears to reduce lung volume, and we see changes in breathing rate associated with that. Before I get into the first example, I do want to give you an overview of how we run studies on the Vium platform and the study design parameters that we use. So what's really important for these studies is that we use standard study protocols for all experiments. So what you'll see laid out in all 3 of these models is that these are the very common ways that researchers will run the models. We don't do anything different or unusual. And what's really nice is that the animals actually remain in the home cage throughout the whole entire study. So at no point apart from when we do the induction and or if we were to give treatments or monitor what we call conventional measures, those are the only times that the animals are actually out of the home cage. Otherwise, all of the data that we're showing you here comes directly through the video cameras and streamed onto the system. So most of these studies include 2 different phases. The first phase is the acclimation phase, and the second phase is an induction phase, or potentially we might also add on a treatment. Each of these phases have important aspects that we look at. So in the acclimation phase, this is the animal's time and ability to get used to the Vium kind of environment. This is standard. This is what any researcher should be doing with their animals, getting them used to the vivarium. Usually that's the first 1 to 4 days of the study. And then between the last 5 and 7 days, we actually take that as an opportunity to collect baseline data on all of the animals. So we'll look at, in its natural state, what is a mouse's general breathing rate or respiratory rate, and how active or inactive are they, and do they show relatively normal circadian rhythms? So this is important as we get to the randomization phase. So as we move to randomizing animals into various study groups, so we might take a cohort of 20 animals and break them up into 5 groups of 4 animals or 4 groups of 5 animals. What we might do is look for animals which have abnormal breathing rates. So if all animals have breathing rates within a small window and we have one animal that is outside of 2 standard deviations, we may actually remove that animal from the study, and that allows us to get more consistent results. And we also make sure that the similar average and standard deviation of all animals are the same for the primary outcome measures that we care most about. So in this case, we focused quite a bit in most of these studies on breathing rate, although we also did use motion and sometimes body weight, as you'll see. And that happens right before the induction phase. So in the final phase, the induction or treatment phase, our goal is to monitor changes in these primary endpoints or secondary outcomes from that baseline state. So we can not only look at group averages, but we can also look at individual animal averages to know whether or not the induction and/or a treatment is having a positive effect. So with that as an overview, let's move on to the first model. So here we'll be looking at a bleomycin-induced lung fibrosis model. This is a model of idiopathic pulmonary fibrosis. So idiopathic pulmonary fibrosis is a complex disease, really of unknown etiology. There are a number of different models available for looking at idiopathic pulmonary fibrosis, but certainly this mouse model of bleomycin, in which the bleomycin is administered directly into the lungs, is probably the best characterized of all of the models out there for testing antifibrotic compounds. There's been a lot of work done to ensure reproducibility across labs. Current best practices for endpoints and evaluation are looking at hydroxyproline. So this is a measure of collagen accumulation. So this will tell you the extent of fibrosis across time. And also looking at histologic assessment of the lungs. Here, there are some studies which look at lung function assessments. They're considered helpful, but certainly technically challenging. And so it's not common practice to actually look at this in vivo lung function in those animals. In terms of thinking about the different phases in this model, which has been extremely well characterized, there are about 3 different phases. So I'll tell you, in our model here, we used male C57 mice. We just will show you data from 2 groups, a control group, as well as a bleomycin group, in which we gave 3 units per kilogram. After a single administration of bleomycin, there's an initial acute inflammation phase, which lasts somewhere between 0 and 10 days. About 3 to 4 days after the bleomycin administration, you start to see proliferation of fibroblasts, and that stage lasts up until about 14 days. And somewhere between 10 and 14, you start to get some pretty severe fibrosis. So keep that in mind as we start to look at the results from our study. So here again, we're using male C57s. We gather during the acclimation phase. We use automated metrics. We look at motion and breathing rate. Conventionally, the only measure that we're looking at here is body weight. And just before disease induction, as I talked about, we randomize these animals based on motion and breathing rate and eliminated any animals with pretty significant increases in breathing rate or decreases in motion. we gave a single administration of bleomycin, and then we're tracking changes from baseline across the next 21 days. At day 21, we euthanized all 10 animals and collected lung weights, hydroxyproline, and we've actually sent out tissue for histology, but that hasn't come back yet. So I will not be sharing that data with you today. So just going through some of the conventional measures to orient you to the graph, and all of the graphs I'll be showing you here are pretty much the same. Along the x-axis, you can see the day of the study, with day 0 being the day of induction, so the day that we gave the bleomycin. The y-axis has the body weight. So you can see prior to induction, the body weights of the animals were quite similar. Following induction, the control animals continued to gain weight. We saw some fluctuations in the animals induced with bleomycin, but none of that was statistically significant. Looking at motion data, so this is how active the animals are. We're showing here average nighttime motion data. Importantly, we look at nighttime motion because this is when the animals are most active, and we find it's most indicative of any kind of an animal feeling bad, or in the case of more CNS-oriented diseases, changes in overall motor function. Again, you can see prior to the dotted line where we gave the bleomycin, overall motion between the groups is quite similar. After that, actually the groups remain fairly similar. And so there was no significant difference between the bleomycin-induced and the control animals. Importantly, this appears to be suggestive that there was a minimal kind of systemic effect of the bleomycin. So certainly if you were to give bleomycin via an IP injection or another method, you could imagine that the animal would have a fairly significant response. So this was quite nice in that it really indicated to us that we were pretty much affecting the lungs only and not more broadly. Moving on to breathing rate. So here you can see, again, prior to bleomycin induction, the breathing rates between all animals were extremely consistent, right around 190 breaths per minute. Once we administered the bleomycin, again at that dotted line, the animals with bleomycin showed a very significant drop in breathing rates, down to almost 120 breaths per minute on average. And that was the initial response. We saw that at days 2 and 3 was that significant drop. And then by about days 9 or so, we saw a significant increase in breathing rate in these animals, which then persisted for quite some time. So again, now we're seeing a short-term decrease in motion, which may be associated with that initial inflammation, and a longer-term increase in breathing rate, which we associate with more of the fibrosis. And just to give you a sense for whether or not this is associated with more of the biochemical indices commonly looked at in the bleomycin model, here we're showing you lung weight to body weight ratios. So here, the animals in blue are those that received the bleomycin versus those in gray received the control. And you can see a significant increase in lung weight. And remember, this is again at day 21. And then on the right, you can see lung hydroxyproline read out. And again, we see a significant increase in hydroxyproline content in the bleomycin animals compared to the control, which would suggest that we do have increased collagen content, and fibrosis. So the question now becomes, those increases in breathing rate, are they well correlated to either of these indices? And in fact, they are. So on this slide, again, I'm showing you the lung weight to body weight ratio, which you saw on the prior slide. And I'm also showing you the correlation now between lung weight and the breathing rate ratio. So here we're looking at the average breathing rate, resting breathing rate on the last day of the study. So this helps you correlate the breathing rate at the time of euthanasia to the lung weight at the time of euthanasia. And here we see a very significant correlation with an R-squared of somewhere around 0.89, suggesting that as breathing rate increases, the lung weight also increases. Perhaps more importantly, we see a similar correlation when you look at lung collagen content. So again, on the left, I'm showing you the same data from before, showing the increased hydroxyproline in the bleomycin animals. And on the right, I'm now showing you the breathing rate to hydroxyproline content. And you can see a fairly nice correlation with an R-squared of 0.65, where again, increases in hydroxyproline content or fibrosis are associated with increases in breathing rate. So the conclusions from this study that we draw is that we have fairly nice signal specificity. So in this case, we're seeing a nice signal in the breathing rate, which occurs around the time of expected onset of fibrosis, but we're not seeing a more systemic response, i.e. we're not seeing any change in motion. In addition, as I just showed you on the last two slides, in both cases, or both measures at day 21, we see that breathing rate is highly correlated to what individuals or researchers consider the gold standard, which is collagen content as well as overall lung weight. And we'll continue to look to see whether we see similar correlations with histology as that data comes in. What I did not say, and what's also important, is we actually think that the signal is fairly, or we can show data that the signal is quite strong. So doing a priori power analysis, so looking back at the study, remember we have Ns of 5 in each of these groups. We discovered that in order to be able to tell a significant difference between the control and the bleomycin-induced animals, retrospective power analysis would suggest that we would need Ns of 3 per group versus looking at hydroxyproline content, which is just a more varied assay. You would need somewhere around Ns of 13, which is actually quite interesting because Ns of 13 or 10 to 13 are what is most commonly used in studies. So what this would suggest is that you can actually use fewer animals in these studies if you use breathing rate as one of your primary endpoints. So the second model that we looked at is an acute pneumonia model. So here we're using bacteria Pseudomonas aeruginosa, which is one of the most common causes of pneumonia, to look at lung infection. So current best practice endpoints for evaluation are removing lungs and actually doing CFU counts, so how much bacteria is in the lung, doing histologic assessment of the lung, as well as monitoring systemic cytokine responses to look at overall response to the Pseudomonas. In this case, lung function assessment is very rarely done, There are only a few papers which actually look at lung function. But in the cases where it has been looked at, the values again correspond quite well with standard endpoints. So it would suggest that if you had an easy way of monitoring lung function, it could be quite suggestive in this model. So looking at our experimental design here, we used male BALB/C mice that were between 9 and 10 weeks of age. And much like the last group, we're showing you data from a control group with an N of 5 and a group with the Pseudomonas aeruginosa, again with an N of 5. We do a standard 7-day acclimation period. The only addition here is we're looking at 2 conventional measures, so body weight and body temperature, which have both been shown to correlate nicely with or be useful measures for looking at pneumonia in these animals. And we're randomizing again on breathing rate and on motion. So to induce the disease, we do a single administration of the Pseudomonas aeruginosa. We gave the animals 5.5 times 10 to the 7th CFU or saline, and we did that through a nasal induction where we did a single drop to the nose and then had the animals inhale the Pseudomonas. And then we track changes from baseline, again looking at the conventional measures, body weight and body temperature for 2 days, as well as some of the automated metrics from the Vium platform, motion and breathing rate. So looking at conventional monitoring of animal health, we see typical signs of illness in these animals. So here we're looking at body weight and body temperature again. So we do daily body weight measurements. Again, you can see that the Pseudomonas in blue, prior to induction at day 0, body weights are not statistically different between the groups. But then by 1 day and 2 days afterwards, you see significant drops in body weight in those animals that receive the Pseudomonas aeruginosa. We also see a very consistent drop in body temperature. So again, the dotted line is when the animals received Pseudomonas. And both 2 and 7 hours post-induction, we see a significant drop in body temperature. But by 24 hours, the animals are back to baseline. So in both these cases, we see signs of illness. In the case of body temperature, we see a transient reduction in body temperature, which is associated, again, with this inflammatory response. Looking at cytokine levels, this is a fairly busy graph, but please take a few moments to look and see. We see significant increases in TNF-alpha, as would be expected, significant increases in IL-10, KCGRO, IL-6, as well as IL-1 beta and INF gamma. And this is a profile which is very reflective of what would normally be seen in this model. And again, these data were taken at 48 hours post induction. Now looking at Vium metrics, here is a graph showing motion data across time, where the dark bars represent nighttime and the white bars represent daytime. You can see prior to induction, which is the dotted line just before the end of the graph, you can see very nice circadian rhythms in the animals with much more nighttime activity compared to the daytime activity. However, post induction with the Pseudomonas, there is a significant drop in nighttime activity. So you can see that the nighttime activity drops about 8 hours after induction, and it actually remains low all through that first night, as well as during the second night. Again, suggestive now that we're seeing a more systemic inflammatory response. When we look at breathing rate, prior to the induction, we have very similar breathing rates. So again, right around 190 average breaths per minute. But within 8 hours post-induction, we see a significant increase in breathing rate, which persists all the way through that first night and into the second night. So this response lasts for upwards of 36 to 40 hours. So next question is, are these increases in breathing rate correlated with the changes that we see in the cytokine data? And the answer to that question is absolutely. So here we're just showing a few examples, IL-6, IL-10, and KCGRO. We are showing the breathing rate is now on the x-axis, and the level of the cytokine is on the y-axis. And you can see a nice correlation with lower breathing rates are associated with lower levels of these cytokines, whereas higher breathing rates are associated with higher levels, suggesting that as you see an increase in this inflammatory response, you see increases in breathing rate. We've also shown this with decreases in motion, but that we're just not showing that data here. So conclusions we draw from this study is that we're getting nice signal specificity again. So again, there's a clear signal both associated with increased breathing rate as well as with a decreased motion now. So this is in contrast to what we saw with the Bleomycin-induced model, and that persists longer than more typical measures of these inflammatory response, in essence, the body temperature. And that again, breathing rate is now correlated to the cytokine levels. So what one could hypothesize is that you could monitor breathing rate and get a fairly nice idea of what the underlying extent of the inflammatory response is. And while I've not shown the data here, I have shown it in prior presentations, and we're doing additional work here. It does seem that this is consistent across a number of different models in which there's a broader inflammatory response. So we've seen nice correlations between breathing rate, motion, and cytokines in an LPS challenge model, as well as an acute liver toxicity model, of which there is data out, and I'd be happy to share that data with anyone who's interested. Again, we also see a nice signal strength here. So looking at retrospective power analysis, it suggests that using breathing rate, we would need Ns of 4 in order to be able to statistically distinguish animals that had gotten the Pseudomonas versus not, with even Ns of 3 for motion, but that you would need larger Ns on the order of 5 to 6 to detect some of these larger changes in cytokine levels. So it does suggest that the signal is a bit stronger than some of the conventional standard metrics that have been used. Okay, now for our last model. We looked at a lung metastasis model using the CT26 colon carcinoma line. So metastasis is the spread of cancer to the non-original areas. So in this case, colon cancer spreading to the lungs. And it's a significant problem that requires improved treatment options. So in this case, in this model, we injected tumor cells from the CT26 line directly into the systemic circulation. And these are some of the most common models for studying lung metastasis. So CT26 is just one line. There are about 10 to 15 lines that researchers can use to study metastasis. Current best practices for endpoints in this case are imaging. So if the cell line is also includes bioluminescent signal, you could look at that, as well as in MRI or potentially using a PET scan to look at the location and the extent of tumor burden in the animal. In this case, we'll focus mainly on looking at gross necropsy and histology, so looking at the extent of tumor burden in the lungs of the animals using a terminal procedure. The model here is a syngeneic model. So we used male BALB-C mice that were 7 to 8 weeks of age. We actually looked at 5 different groups in this model. So we used a control IV injection. We gave 2 different concentrations of cells, a 1 × 10 to the 4th and a 1 × 10 to the 5th, both via IV injection. And to show that it really is tumor burden in the lung and not associated with the animals having these cells injected more broadly, we also did a subcutaneous flank injection. So we have a control group where we did a subcutaneous injection of saline, and then a 2 × 10 to the 6th of the CT26 cells injected. As always, we acclimated the animals for 7 days, and then we gave either that single IV or subcutaneous injection of the CT26 for a control. And we tracked changes from baseline. Here we used body weight as the only conventional measure, as well as changes in motion and breathing rate. And to look at tumor progression, we euthanized 2 different groups of animals, one at day 21 and then one at day 28 post-induction, to look at the extent of tumor formation in the lung as well as in the subcutaneous group. We looked at the size of the tumor on the flank. Looking at body weight, in all of the graphs on this, in this section, I'll show you the IV and the subcutaneous tumor groups graphed separately. So here you can see that in the lung metastasis model, prior to induction, body weights are very similar, and they remain very similar post-induction until about days 24 post-induction, where we start to see a very significant decrease in the animals that received the high dose, but not the low dose of the CT26 cells. So those animals in the gray are those that received the saline injection. The light blue are the low dose of cells, and the dark blue are the high dose of cells. In the case of the subcutaneous tumor group, we actually see increases in body weight in the animals that received the subcutaneous tumor. This makes sense because the tumor weighs something, and therefore those animals are getting heavier. We start to see changes around 20 days after the injection, and it becomes more statistically significant the farther out you go. The next question was whether or not we actually saw metastases in the lungs. So here I'm showing you control animals in the first column. The second column are those animals that received the IV injection. The third column are those animals that received the subcutaneous injection. So those are the injection on the flank. The top row, my apologies that that's not labeled, are example lungs from day 21. And the bottom row are example lungs from day 28. What you can see in the control animals is that we really see no metastases. We also see none in the subcutaneous tumor group at either the 21 or the 28 days. And then in the IV group, we see very few in this example, perhaps one metastases at one nodule at day 21. But by 28, you can see the lungs are filled with these tumor nodules. We went on to quantify those. So here I'm showing you nodule count on the left. So here we filled the lungs with India ink to allow the nodules to stand out a little bit more. And what I've graphed are the number of nodules, both at the early and the late time points for each of the conditions. So what you can see in the IV low dose cell line, we see very few nodules. It's on the order, I believe, of 3 to 4 on average for both the early as well as the late time point. In contrast, for the animals that receive the high cell count, we can see a significant increase both at the 21-day time point as well as at the 28-day time point, with a significant increase between the 21 and the 28-day time point, in contrast to what we see in the IV saline injected animals, where we see no nodules at all. On the right, just to show you tumor size, we actually did conduct caliper measurements on the animals with the subcutaneous tumors. And there you can just see that in comparison to the control animals, which are in gray and had no tumors, that we did see significant increases in tumor size, or we did see significant tumor size in those subcutaneous tumor injected animals. So now looking at the Vium metrics, we saw significant changes in motion, but only in the lung metastasis group. So on the left, again, we see the lung metastasis group. And prior to injection of the tumor cells, motion was very similar across all groups. Very similar to the body weight, we see no real change in motion until about day 23 after injection, at which point we see a significant decrease in the motion in just the high cell group. In comparison, there was no significant difference in the animals that received the 1 × 10⁴ cells. Looking at the subcutaneous tumors, there was no change in motion at all in those groups. Both groups showed similar motion profiles. Now we're looking at breathing rate. And again, similar to what we saw in motion, we only see significant changes in breathing rate in those animals which had tumors in the lung, so the lung metastasis group. So prior to injection of the tumor cells, you can see that the average breathing rate is somewhere around 180 breaths per minute. This is very similar across all of the studies, in fact. In those first 20 or so days, we see no real significant change, although perhaps the low cell group has a few days in which they have significantly higher breathing rates. But once you get out to the point where we start to see tumor nodule growth in the lungs, those animals that received the high number of cells have a very significant increase in breathing rate in comparison to the animals which had the low group, the low cell number, which showed an intermediate increase in breathing rate, all of that compared to the control animals whose breathing rates really did not change across the course of the 28-day study. So here we can see a dose-dependent increase in breathing rate in these lung metastases groups. In comparison, again, remember the goal of the subcutaneous tumor group was to show that changes in breathing and or emotion were not associated with just having the cells in the animal's body. Again, you can see that in this group, we see no significant difference in breathing rate in these animals across the course of the 28-day study. So the next question is, it seems convenient that we see increases in breathing rate in those animals with the high nodule count versus the low. Are those significantly correlated? So here, what we're showing you are average breathing rate. Again, this is at the terminal day. So we're looking at only breathing rates at the time where we can actually have a good correlation with lung tumor nodule count. And we're comparing that to lung tumor nodules. And you can see, again, a significant correlation here between breathing rate and lung tumor nodule count with an R-squared of somewhere around 0.65 or 0.66, suggesting that as the lung becomes more burdened with tumors, it becomes more challenging potentially for that animal to breathe, and breathing rates are now going up. So from the lung metastasis model, again, we feel that we are getting signal specificity here. So we have a clear signal, again, in motion and in breathing rate for those animals that have lung metastases, but we're not seeing a signal in the mice with subcutaneous tumors only. What's interesting is that decrease in motion, which I showed you, precedes the significant decrease in body weight, which would suggest that it's a little bit more sensitive than body weight for looking at overall animal health in the case of animals with lung metastases. And finally, that again, breathing rate is highly correlated to the number of tumor nodules in the lung. So you can imagine with additional tumor nodules, you could imagine the breathing rate may continue to go up, and that this is potentially a very sensitive way to be able to monitor or to get a sense of tumor burden in animals without having to euthanize them at specific time points and or do potentially more invasive monitoring of animals using PET scan or MRI. So summarizing the conclusions from all of the use cases here, so the overall question that we asked was, can automated monitoring arresting breathing rate actually predict lung function in these models? I've showed you data from three different models, one from an idiopathic pulmonary fibrosis model, a pneumonia model, and a lung metastasis model. And again, we looked at each of these models because you can imagine a different underlying cause for each. And we wanted to see how consistent it is across each of these different kind of mechanisms. We showed increased breathing rates with fibrosis that correlate to collagen content in the idiopathic pulmonary fibrosis model. We showed increased breathing rates and decreased motion associated with systemic inflammation, and that breathing rate correlated with cytokine drivers of the immune response in the pneumonia model. And finally, we showed that breathing rates increased with increased tumor burden, but only in those animals with lung metastases, and that those breathing rates correlate with the number of nodules in the lung in that CT26 colon cancer metastasis model. So thinking about the conclusions from these studies, we go back to the question that we asked at the beginning of this presentation. So remember, the question was, plethysmograph and forced oscillation are using the flex event. Is there potentially another way of monitoring lung function by using resting breathing rate in this automated system? So we asked the question, were the results consistent? And do they recapitulate underlying biology and or pathology in the animals? And what we hope we've shown you today, or given you a taste for today, is that the answer to those questions are probably yes. So what we're showing you here is that using an automated home cage monitoring system, where in essence all you have to do is run your study in your typical way, by being able to measure resting breathing rate in these animals, we're measuring, in essence, a correlation of lung function within freely moving animals that are truly in their natural state. So while the plethysmograph is more in their natural state, you still have animals which are constrained and may show stress responses in comparison to animals that are in their home cage environment. And we actually think that because they are in their natural state and they're not stressed out and they're not being interacted with by humans, that this is actually why we're getting far more conclusive results using the home cage system versus a plethysmograph as an example. We also show that we can do continuous monitoring, which allows for kinetic assessment. So rather than taking the animals out and doing many different time points across, in the case of a plethysmograph, or perhaps having to have many different groups in the case of the flex event system, if it is a terminal procedure, by monitoring 24/7, you can actually see the increase in breathing rate kind of on an animal by animal basis to be able to get an assessment from each animal about potentially how sick, what that is, systemic inflammatory response is, tumor burden, et cetera, because we know that not every one of these models follows a kind of perfect kinetic path. So overall, we think that this is potentially a new way in which you can actually monitor lung function across a number of these lung disease models to get a fast, easy, and fairly accurate assessment of underlying biology. And with that, I open it up to any questions, and thank you for taking the time to listen today.
Thank you, Dr. Schaevitz, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the answer question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, in what other types of therapeutic areas or disease models do you think that breathing rates would provide additional insight into disease?
That's a great question. So I alluded in the presentation to one area where we found breathing rate to be quite informative, and that's in any form of systemic inflammatory response. So we've looked across a number of different responses and seen changes in breathing rate associated with the LPS induction or challenge model as an example, acute liver toxicity. We've also seen changes in breathing rate in a number of models in which individuals would not necessarily expect to or think about breathing as being a key primary outcome measure. So we've seen it in a number of models of CNS and neurological disorders as well. So I think the answer to your question is it's probably broad, and we could possibly see it across a number of different therapeutic areas. We generally take the approach of the platform reads it out on all models, and therefore we should be able to get a sense across models of which ones might be potentially most interesting.
Our next question is, does this system increase your study power?
Again, a great question. So I tried to go through quite a bit of that across the presentation, But we are seeing that in many instances, being able to monitor these metrics basically 24/7. So we're getting, I didn't actually go into this in the actual presentation, but we get somewhere around the 2 million data points for motion, and we're getting somewhere much fewer in the way of breathing rate points just because of the fact that we have to measure resting breathing rate. But because we're getting them both day and night, we actually see that we can use fewer animals potentially in order to get very high power. I think I showed you in the bleomycin model that we would need Ns of 3 or 4 in order to be able to statistically distinguish the groups versus Ns of 13 with more traditional measures. So again, it'll depend on a study by study and a model by model basis, but in general, we're finding that being able to use these continuous measures, we do get increased study power.
Now, Laura, this is a 3-part question. Interesting data, thank you. If I wanted to run a study on this platform, what are my options? Do you sell the system? And do you collaborate to develop new models?
So the answer to your question is yes and yes. So we've just recently started to sell a production version of our system and have actually just recently done a few installs. So it is available for those individuals who want to go that route. We're also very interested in collaborating with groups of individuals who have models they think would be highly conducive to the Vium platform. So we work with a number of academic groups as well as pharma, biotechs to develop models and to identify the best metrics and additional metrics that they could potentially use. So, feel free to reach out to us on either of those if you're interested in actually working with or running your models on our platform.
And it looks like we have time for one more question. You mentioned these are resting breathing rates. What is the frequency of breathing rate measurements you get?
Oh, great question again. So we've actually looked, one of the fun parts about our system is that we can look at all of the data that we've collected over the last actually 4 years. So we've looked at all animals, rats, mice, and whatnot. We find that on average, we get about 150 to 200 data points for mice. The majority of those actually occur during the day when the animals are asleep because they tend to be at rest more. In rats, that number is actually a bit higher. So we get somewhere, although I didn't talk about any rats today, but we get somewhere around 300 or so breathing rate points over the course of a day. So you can imagine, depending upon the length of your study, how many data points that might be.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of the registration. We would like to thank LabRoots and our sponsor Vium for underwriting today's educational webcast. This webcast can be viewed on demand through December of 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.